So welcome to our corporate workshop on clonality assessment and monitoring using our LymphoTrack next-gen sequencing products. We really have a good workshop for you today. I'll do the introduction. I'm the founder, chief scientific officer of Invivoscribe and the companies. Then Dr. Michael Klass will walk you through the next-gen sequencing products, some of the validation and analytical assays that were done, or some of the studies that were done to validate the assays. And then we have really two excellent guest speakers. So Dr. Rina Kansal, from Hematopathologist from my alma mater, UCLA, and Maria Arcila, who's the acting director of the Diagnostic Molecular Pathology Lab at Mememorial Sloan Kettering Cancer Center. So on that, I will turn the mic over to Dr. Michael Klass, who's our Senior Director of Product Development.
I'm going to talk about how Invivoscribe is using next-gen sequencing for clonality testing of lymphoproliferative diseases. I'm going to give you an overview of our technology improvements. We've been very busy during the past year. I'll talk about our workflow, our menu expansion, the software that we provide with each kit for the analysis of the data. And I'll describe how we have developed tests on both platforms, the MiSeq and the PGEM, as well as their assay performance. Throughout the development of these assays, we've been primarily focused on the ease of use for the customer. Simple workflow. We concentrate on a one-step multiplex PCR for library construction. In other words, you don't have to do 5, 6 different PCR reactions. We provide the barcodes in the primers themselves, so it's one multiplex PCR. We provide primer combinations that target all functional rearrangements, and we provide that in a flexible workflow through availability of multiple indices. As Jeff said, there's 24 indices that you can run for the MiSeq and 12 indices that you can run for the PGEM. We provide the complete solution. We also provide the software for simplified data analysis, because many of you said, I don't want to send you my samples, I want to run them myself, I want to keep total control over them, and I want the data to be analyzed in my hands, and I want to see that. So we provide that. Our available assay targets are TCR gamma, IGH, IGH for somatic hypermutation, and in development we have TCR beta, as well as IG kappa. We can run both IGH and TCR gamma on the same platform with the same indices, and we provide you with the software that does the analysis and visualizes all the data and separates that information for you. Let me give you an example of IGH assay run on MiSeq. We had a total number of reads of 413,000. You click on this cell and you can see the exact sequence of the most abundant clone. You get the length, you get the raw count, you get the V gene assignment and the J gene assignment, as well as a % total reads. So let's talk a little bit about somatic hypermutation. In our talk last year, people came up and said, okay, I like this test, but I need to do the somatic hypermutation analysis. Why? Because somatic hypermutation, which is defined as greater than 2% difference from the germline of the IGH region, is important for prognostic value. Failure to undergo somatic hypermutation indicates a more aggressive disease. Also, just the presence of the V3-21 region indicates a more aggressive disease. That information, that sequence information, obviously you can get from next-gen sequencing from our assays. The current method of determining somatic hypermutation often involves PCR clonality detection, as well as cutting a band out of a gel, extracting it, sending it off for Sanger sequencing, getting the Sanger sequence back, and doing the IMGT analysis. We automate all of those steps for you. You don't have to do that. So let me show you what you get with the somatic hypermutation. Here's an example, IGH sample that we ran. We got a total of 464,000 reads. You see here we have somatic hypermutation, which is the % mismatch to the reference gene. We use the reference gene set IGBlast, and it correlates very, very well with the results from IMGT. In addition to that, we also provide information on whether or not that sequence, the clonal sequence, is in fact in frame. We do all of this analysis because we recognize that it's important for determining somatic hypermutation rate, as well as whether or not that clonally expressed sequence is actually expressed and is in frame and has no stop codons. All of that's important for somatic hypermutation analysis. Now, in terms of making it easier and more economical for the customer, is we have done combinations of assays. We can run 4 different targets, all on the same flow cell, same MiSeq flow cell or PGEM, with the same indices. For example, here you see the result of 96 reactions run on the same MiSeq flow cell. Now, the other thing that you said was important when we talked to various customers was minimal residual disease testing. This graph is a representation of the data from a Lancet article. It shows the leukemic burden and shows the importance of keeping that leukemic burden low. Low leukemic burden indicates good prognosis. High leukemic burden indicates failure and early relapse. So what you want is you want a technology that is the most sensitive to detect the leukemic burden, because you want to drive that leukemic burden all the way down to undetectable levels. And I'm going to tell you that next-gen sequencing can get down to 10 to the minus 6, or 1 in a million cells. But enough from me. What I really want to stress is some of the results from some of our early adapters of these tests. And I would like to introduce now Dr. Rina Kansal from the Department of Pathology and Laboratory Medicine. Dr. Kansal is a hematopathologist and is going to tell you about her experience with these assays.
So I wanted to look at T-cell clonality first, because it is much harder to assess clonality in T cells than in B cells. So this is the assay that we evaluated, LymphoTrack TRG assay for the MiSeq. We studied 41 archived samples with a prior diagnosis based on integrated clinico-pathologic information. I'm a hematopathologist, not a bioinformatics person, but this software, I could handle it. And once I learned how to do this, it was as easy as working with any other software. So our results, we succeeded in all our library construction and sequencing in all 23 blood and bone marrow aspirate specimens and 18 FFPE tissues. So I'll share with you some of our sequencing library QC data. A Q-score of 30, as you all know, is 99.9% base call accuracy. And our data is, the majority of it is at Q40, which is really 99.99 base call accuracy. So I'll share with you some of our cases. First case, I start with a benign lymph node. I'm a pathologist. I need to first understand the normal before I can say what is atypical, what is lymphoma. This shows you one of the views that Mike just showed. The software gives this to you. The top 200 rearranged VJ sequences. And as you can see, this data is based upon 286,000 something reads. And all these little colors, each of these represents a unique sequence, a unique rearranged sequence. And that is a typical polyclonal VJ rearrangement picture. Then another case from a patient with an enlarged inguinal lymph node who has a peripheral T-cell lymphoma. This is the PCR TRG 2.0 assay. This shows the, again, 2 clonal peaks. This is about a 10 nucleotide difference, and that's what you see here. Now here, there is only one VJ region that is rearranged. However, this one and this one, there is a base pair difference of 10 nucleotides. So again, very consistent with what the PCR is showing. One thing we definitely learned is that this sequencing assay worked very well in our hands and in our lab for both fresh and FFPE tissues that were routinely submitted. And this analysis definitely provides much more objective, specific, and accurate information in comparison with PCR. And with that, I just want to acknowledge the Department of Pathology and Laboratory Medicine, everybody here who helped tremendously with my project. And I will introduce Dr. Maria Arcila.
Thank you so much. Whenever I see this data, I get so excited because we have learned so much about disease by just looking at these tracings and looking at this massive amount of information. So my goal for today is to just take you to what our experience has been in looking at these samples. We have completed our validation of TCR gamma and IGH. We're looking into our validation of the somatic hypermutation assay. And we have been using the assay whenever we see a case that is difficult. We would just go ahead and assess it in addition to the fragment analysis. We'll do the NGS assay. PCR-based clonality testing is an essential component of the workup of lymphoproliferative disorders. But when you get something like this, what do you call it? It's a little bit more difficult. So this is where I think the NGS assays have really helped us in many of the cases to be able to make these calls of clonal versus non-clonal. So why look to gene rearrangement testing by next-gen sequencing? Well, I think after looking at all the cases that we have done, that it is a very valuable method for the initial evaluation of lymphoproliferative disorders. It allows the identification of the full range of the clonal populations that you see in a specific sample. It determines the DNA sequence of that clonal rearrangement, and then it detects clonal events that may be hidden in a polyclonal distribution that you cannot possibly do with a fragment analysis. You can also track residual disease, and for the B-cell processes, you can examine the somatic hypermutation as a prognostic marker, which we would do for CLL, and now there is emerging evidence in hairy cell leukemia as well for the somatic hypermutation. But beyond the detection, you also may identify in the samples both stable and dynamic aspects of immune repertoire that differs under normal conditions and under disease conditions. It provides a very high resolution picture of the spectrum of immunity that you wouldn't see otherwise in that lymphoid malignancy if you don't do something like this. And it also defines the initial behavior of clonal tumor populations. And you can see how these clonal populations are suppressed or reemerge when you look at new samples. Now, this is case number 2. This is a 57-year-old female with a history of CLL, SLL, who then comes with an enlarged right axillary lymph node that by morphology looks like it was 70% diffuse large B-cell lymphoma and 30% CLL. So the morphologic diagnosis was this is a patient with history of CLL who now has Richter's transformation. There is no prior clonality testing in-house for comparison, but in the chart review, we did see that this patient did have a hypermutation, a somatic hypermutation status that was in the chart. And you can see here that the rearrangement that the patient had was a V459, and it was a productive rearrangement with a mutation frequency of 0.7%, so it was said to be unmutated, or the somatic hypermutation status was unmutated. So this is the fragment analysis you can see here, so it actually has 2 clones. This is the current sample. We didn't have anything from the prior sample to be able to know. So clearly there is something here that is more than just this. So we did do the NGS assay, and what you can see here is that this patient has, in fact, 2 clones. One of them is the rearrangement, the 459, and you can see it here. And then, the dominant clone, which is this one, is a completely different rearrangement. It's a V348. So the question, and this raised the question, is this actually a Richter's transformation? Because it's a completely different clone. There is no relatedness here. So is this a patient that has CLL and now has a B-cell lymphoma, but they were found in exactly the same lymph node? So the treatment for something like this is completely different when you say it's a Richter's transformation or a patient with CLL who now has a completely unrelated B-cell lymphoma. And now this is case number 3, and this is the CSF that I showed you in the validation before. This is a 67-year-old female with history of CLL, another CLL, and that history was since 2002. This patient presented in 2010, not with a CLL, but with a mantle cell lymphoma, so an additional malignancy, And we knew that it was, in fact, a mantle cell lymphoma because it had co-expression of CD20, CD5, and cyclin D1, and by FISH, it had a confirmed translocation of 1114. Now, this patient shows up in 2014 with a headache. Now, what does the NGS tell us more about this patient? So this is what we actually see. So what we see here is that these are the clones, and all of the clones are exactly the same size, but they're not exactly the same clone. So each individual population here is 1 or 2 bases apart. So even though you have exactly the same fragment, what you have is multiple and multiple populations that are related. So we interpreted this as mantle cell lymphoma, leukemic phase with CNS involvement, and this is suggestive of clonal evolution. So there is a lot of interpretation that goes along with having all of this information that goes beyond what you can possibly tell by a fragment analysis assay. Now, case number 4 is a 57-year-old male with a growing subcutaneous nodule in the arm. This patient was given the diagnosis of an atypical T-cell proliferation that was most consistent with an angioimmunoblastic T-cell lymphoma. So immediately after the patient was diagnosed, the patient gets a staging bone marrow, and the staging bone marrow was completely negative for disease. Morphologically, by flow cytometry, completely negative. However, the clonal study, if you do it by fragment analysis, you do find that these two little clones are exactly the same in here, but in a polyclonal background. So this may be difficult to interpret as exactly being involved by disease. But when you do the next generation sequencing assay, you actually go and look for the specific clones and the sequence of that clone, and you do find that those 2 clones are exactly the same as these 2 clones in here. So the patient gets treated for residual disease now based on the NGS assay, and then you have a post-chemo bone marrow, again done before transplant to figure out if the patient has any disease, and the patient is completely polyclonal, and you cannot find those sequences anywhere in here. So you can see here the many times that we did this same assay with that sample, and again, you can see that the reproducibility from run to run is very, very, very good, and then we cannot find those 2 clones anywhere in here. So patient is free of disease based on that last sample that we got. Now, we also played around with this, with the assessment of somatic hypermutation, and as you know, this is an important prognostic assessment of patients with chronic lymphocytic leukemia. And the gold standard is the PCR capillary electrophoresis-based assay that then you have to Sanger sequence, you have to separate the clones, you have to clone a lot of the times, and this is very, very labor-intensive and time-consuming. From the perspective of a lab like ours, do I really want to be doing that every day? The answer is no. So an assay like this is actually time-saving, and it just makes for a much better workflow in a laboratory. So these are my conclusions. I think that the assay very efficiently detects IGH and TCR gamma gene rearrangements using a single multiplex master mix. The results are highly, highly concordant when compared to fragment analysis and Sanger sequencing. And it concurrently identifies the sequence information that we require to be able to track these clones in subsequent samples, which is life-saving for a lot of these patients. The somatic hypermutation analysis is simple and highly concordant. I wouldn't go back to cloning if I had to. I would definitely stop doing it, but so the assay works really well. And the MRD assessment level, we actually have not established what the limit of detection is for us yet. So we have a lot of work to do in terms of the limits of detection and the MRD level. But of the samples that we have done compared to flow cytometry, we can go beyond the 1 times 10 to the minus 4 that the flow cytometry is given, because we have had cases where we actually pick up the clone beyond that on a negative study by flow cytometry. So I would like to thank all of them, and also thank Invivoscribe for the very, very invaluable technical support that they have provided us as we have validated this assay.
I would like just to take one more second to thank both Rina and Maria for an absolutely wonderful presentations. A lot of work by a lot of very talented people in the laboratories. Thank you.